Can you hear me, or should I use the microphone? Can you hear it?
Yeah.
No. Thanks again, Nina, and thank you for coming here. Possibly created the But biology is two things. It is an experimental field of research, and it is a new theoretical field. Right? Let's keep the two things famous. Experimental is the experimental research for all codes that exist in life, and the theoretical is the Today there are two dominant paradigms in biology. One is the chemical paradigm, the idea that life is chemistry, or more precisely an extremely complex form of chemistry. And the other is the information paradigm, the idea that life is chemistry plus information. Now, these two paradigms coexist because none is managed to displace the other. Basically, biology is split in two. Either life is chemistry or there is some chemistry and information. So the proponents of the chemical paradigm have not been able to explain why informational processes like heredity and natural selection do not exist in the world of chemistry. And the proponents of the information paradigm have not been able to prove that biological information is ontologically different from chemistry. Now, the discovery of the genetic code, on the other hand, has brought to light a third key component of life, because a code is a set of rules that gives meaning to something, and meaning is different from both chemistry and information. So the existence of the genetic code, in other words, suggests a third paradigm, which states that life is chemistry, information, and codes. And so let's see. That's it. That's the second one. So that's the point. Molecular. That's the point. Now, the origin of the genetic code was the origin of protein life, which is life as we know it. And yet, codes are not mentioned in the two dominant paradigms. And that's the problem. Why? Why is it our present paradigm do not regard codes as fundamental components of life? That's the first point here. Now, in order to address this problem, let us begin from the beginning and see what the code is. In the genetic code, the term code was chosen because there is a similarity with some familiar codes. The Morse code, for example, is a set of rules that creates a match between the letters of the alphabet and groups of dots and dashes. Now the genetic code is the Morse code. So apparently, similarity is there. Object of two different words and the rules to connect each other. So, what is essential in a code? And we have to answer this question in order to see, to find out if the genetic code is a real code or not. What is essential in a code is that the coding rules could have been different. In the Morse code, for example, any letter of the alphabet could have been associated with countless combinations of dots and dashes. And the present Morse code was derived by selecting a small number of these combinations. And this is what defines all codes. A code with a small set of rules selected from a potentially unlimited number in order to establish a specific correspondence between the object to independent code. That's an important point. A code is a code if the rules are potentially unlimited. If we want to prove that the genetic code is a code, we have to prove there was an unlimited way of realizing it. Now, in fact, the existence of the code has the fact that the rules of the code would have been different means that they obey the laws of physics and chemistry, but they are not determined by them. And this is expressed by saying that they are arbitrary. That's the meaning of arbitrary rules. Rules that could have been different in an unlimited number of ways. In fact, that is what it is precisely the presence of arbitrary rules that the two dominant paradigms have not accepted. This is why the chemical paradigm and the information paradigm have not accepted the code as a fundamental, even the genetic code. In fact, according to the stereochemical theory, the rules of the genetic code were determined by chemistry. And if they were determined by chemistry, it means that the code is not the code. The genetic code is just a metaphorical code. It's not a real code. That was the core of the present paradigm. The genetic code is not made of arbitrary rules, so it is not a real code. The rules are determined by chemistry. Okay? And this is why the term code does not appear in the present paradigm. So, in fact, since 1954, George Gang proposed the stereochemical theory. Then there have been many variations of this theory for about 50 years, and different ways of, they found out that the stereochemical theory predicts that. So let's see if it is an empty root false. So let's try again. Let's make another version. Let's say if this root false. And then try again, and so on for 50 more years. And the answer, okay, passed. That's it, that's enough. The point is that we do have the proof that the rules of the genetic code are arbitrary. Because it is possible to just change a few molecules in the data, and you have different rules. Anyway, as I said, remember this. For more than 50 years, biologists believe that the code is not made of arbitrary rules, it is the code. Now, the humanists from the human world want to be, oh, look at that, what a wonderful thing. There is a code in biology, there is a code in nature. And biology, you fool. I mean, you don't understand. Well, that's the situation which has gone on for so many years. So this is a strong message of the stereochemical theory. The genetic code is not a real code, it is only a metaphor, a name which has been adopted simply because it was intuitively appealing. There was similarity to the Morse code. So that's the point. A code is a code if it is an unlimited number of possibilities. We can, to any letter of the alphabet, we can switch whatever number of combinations. And that must be true for this. If it is true, then the genetic code is a real code, otherwise it's not. Okay. Now, in protein synthesis, the sequence of codons is transferred into the sequence of amino acid, and the bridge between them is realized by a third line of molecules, which is adapted, which is the transfer area. Now, you can see here, the adapters, transfer RNAs, are molecules that perform two distinct operations. In one hand, they recognize the codons, and at the other hand, they recognize the amino acid. You can see there is a distance. Now, the problem is,
if we try to change something in between, let's see if we get a different rules. And this is precisely what has been demonstrated by many laboratory experiments, starting from 1987 and so forth, Schumann and so forth. For example, we introduced 2 extra nucleotides into the transfer RNA without changing RNA codon, and found that the resulting RNA was carrying a different amino acid, different rule. And that is being done in countless different ways. Just change molecules there, and you have a different rules. The rules of the genetic code are potentially unlimited, exactly like the rules of the Morse code. Okay? Despite these experimental facts, the stereochemical theory went on and on, and people keep trying to prove it in different ways. Basically, biologists do not believe that there are arbitrary rules in nature, and that's the core point. The genetic code must be determined by chemistry. And yet, all the experiments has changed something in between there, and you have another rules. So we know, we have the experimental proof that there is a potentially limited number of rules in the genetic code. So it is a real code. Right? And the problem is that once you've chosen the rules, you must conserve them. And remember one thing, the only thing which has been conserved for almost 4 billion years since the beginning is the genetic code. Everything else has been changed, but that is not that. Okay? So that's important. Not only you have the genetic code, but that's the only thing that life might conserve. So that tells you that it's at the center of life. Life is there. If you change that, you don't have life as we know it. Anything else can change. Right? So, not only is the real code is made of arbitrary rules, but once you've chosen them, you want to choose a set of rules, you cannot change it anymore. Otherwise, you have a different important words, a different kind of life. So, these and many other experiments have proved that the rules of the genetic code can be modified in countless different ways by adding and subtracting a few molecules. This means that there is a potentially limited number of coded rules, and therefore that they are arbitrary rules. Okay? Remember this, the term arbitrary means that there is a potentially unlimited number of rules. And then you have to choose one. That's the meaning of the term arbitrary. Right? Okay, so we have the experimental proof that the genetic code is made of arbitrary rules, is therefore a real code. Now, next step. According to modern biology, there are only 2 codes in nature. The genetic code that appeared at the beginning of life, and the cultural code that came for almost 4 billion years later. Okay? Again, another of the top concept. Genetic code at the beginning, cultural code at the end. Nothing in between. Right? That's biology today. Now, in fact, many other codes have been discovered at the beginning, but none of them is in any biochemical book, in any biology book. They exist, we have the proof, and they are not in any university book. They do not exist for the students. Okay? That's the reality. I mean, take any biology book and tell me if they mention single transaction code, splicing code, whatever. None. Okay, so this is that's the reality. I usually put a black square in between this, so this is not. So this is what is a code? So a code is a set of arbitrary rules. Arbitrary, remember the meaning of one thing. establish a correspondence between the object of two independent models by means of the third parallel object called a matrix. So, in order to prove in the lab that this is the code, we need to propose three things. Two independent worlds of object, that the apparatus is probably mapping between them, and that the mapping rules are arbitrary. If you prove these things, you prove that there is a code there. Okay? So let us see an example of another code. And in particular, let's start with I want to give you just two examples of the codes that are different from the general code, just to show you the reality of the codes. It means to prove in the laboratory that codes exist. All right? I mean, you don't have to take my word for it. I mean, you just look at experimental proofs. Okay? That's what I'm necessary to read. Okay. Signal transmission and signal transduction. Now, the signals that comes from the environment on the cell, they go into the cell, somehow they reach the nucleus and produce a response. So how does it happen? In the old days, it was thought that a signal from the environment simply crosses the membrane and reaches the nucleus. That's transmission. What we call transduction is another phenomenon, which is that the signal from the environment go on to the membrane. In the membrane, they are changing to a different signal. So the first signal in the elementary are called first messages, and the second one are called second messages. Signals must be changed into a different form. And what I'm going to say is that what makes the change is a code. Okay. So the first characteristic of signal transduction is there are hundreds of first messages, but only 4 families of second messages. This shows that first messenger and second messenger belongs to 2 independent worlds. Right? That's the first condition. You know? We need object from 2 independent worlds. In fact, that is the hundreds of first messengers, 4 families, the first message and 4 families of second message. That's the reality of signal transduction. Now what we know is this. This is transmission from first message to second messenger. It must follow rules. What kind of rules do we follow? Rules. Then there is no deterrence between first message and second message. The major problem of signal analysis is to understand that rules are deterministic model. The answer is the fact that the same first messengers can activate different type of second messages, and that different first message can activate the same second message. For example, acetylcholine is a signal that the brain sends to many muscles, but the skeletal muscle reacts by contracting, the cardiac muscle reacts by relaxing, another muscle does react forward. Another example is that the first, there are many different first messages that release the same second messages in the form of calcium ion. So, these and many other examples. I'm sorry about that black ball, but you see, I used to say sometimes I used to use it to cancel it, to speak without having a slide. And then now I have to go continuously from one to another, so that it is not correct all which should be in the first place. Anyway, that's it. So, seeking instruction, it would be that any first message that it goes to be exactly the same second message. It's received the message. In fact, what we have the same first message can be associated with different second message, and different first message can be associated with the same second message. So you have all possible combinations between them. And only that, so you have a potentially limited number of links between the first message and second message. And then finally we have, here you are. You have the proof here that the adapter, the molecules that make the transfer from first message to second message, is an adapter. Which means that it rules out the rules of the code. So the first message is received by your receptor, and there is an mediator, an amplifier, and then you have the second message. And then you can do that in countless different ways. So the membrane receptors are two adapters. This has been proved by the fact that those receptors follow three independent paths: one that receives the first messages, one that produces the second message, and the third part that can associate the first messenger to a second messenger in many different ways. Okay? So the rules are potentially limited, and what is chosen has been chosen according to the classical English code is correct by choosing a small number of rules out of the potential number. So in signal transduction we find all three characteristics that define a code. We find two independent worlds of object, first messengers and second messengers. We find that there are adapters, provide a mapping between them, and we find that the mapping rules are arbitrary. There is a potentially limited number of signal construction rules, and we can prove it. It's not. There's nothing there's the strength from the theory for we conceived idea and so forth. All these things are proved in the world. And that's how they prove that real biological code exists in nature. Another important example is the splicing code. I will give you these two examples: signal protection code and splicing code. To give you an idea what it means to prove of the experimental proof of signal protection, splicing. About splicing, all genes are copied into RNA molecules that are called primary transcripts. And all proteins are synthesized by RNA molecule called messenger RNAs. Now in prokaryote, the primary transcripts are directly used as messenger RNA. But in eukaryotes, things are more complicated. In eukaryotic cells, the primary transcripts are first cut into pieces. You can see that. They are cut into pieces on the first hand and the second hand, and then the two hands are put together in the center. The primary functions are cut into pieces. Some of them called introns are removed, and the remaining pieces called exons are joined together from the rest of the animals. Now, this cutting and sealing operation correctly removes the splicing, and are realized by huge molecular machines called spliceosome. Spliceosome is actually almost as big as a ribosome. It's a big machine. And the adapters are the small RNAs, which are equivalent with transfer RNAs of the genetic code. There is a bit of a correspondence there. There must be, the logic that have a machine that performs an operation in many different ways. Now, this cutting sequence is splicing. Okay. Now, the small nuclear RNAs are true adapters because they perform two independent evolution processes. One from the beginning and the other from the end of the intron. You must recognize the two ends of the intron. But the important point is that the choice of the beginning of the intron is completely independent from its solid center. To any beginning, you can associate any ending with the cutting operation. So, which means that there is a potential and limited number of relationships between them. And this is true the rules of splicing and arbitrage. So, again, we find two independent words, primary transcripts and messenger RNAs. We find that the adapters provide the mapping between them, and we find that the mappings rules are arbitrary. There is a potential limited number of them. So, on top of that, in splicing we actually have another complication of that, which is called alternative splicing. The fact that from the same messenger RNA, if you don't have a protein, you can have an alternative proteins. And actually, during embryonic development, this can be changed. So, the rules of the splicing code change during development. And there is an example in this course, this tells you how it works, the splicing. That's the concept of identity splicing. You have a sort of sequence of codons there, and then you have some rows which changes those letters. And if you change certain rows, you have this final result. If you change those rows, you have this one result and so forth. You can change them in any way you can. And you can prove in the lab. You can change those values and you can obtain whatever. To any end of the intron, you can associate any other meaning. And that means those are the messages. You can get whatever message you want from them. Okay? That's it. Okay, so far? So what I wanted to know is this. If we want to prove in the laboratory that the biological code exists, we must prove these things. Two independent worlds, the adapters and the antitribes. The fact that the rules can be done in a potentially limited way. When you have those conditions, we have the experimental proof that we have a biological code in progress. Okay? And this is the main point. Despite the fact that biology do not believe the proofs are there, you will never find signal transduction code in a biochemical book. Never. Because they started signal transduction before the discovery of the genetic code, and they have never used the word code for signal transduction, despite the fact that we have the experimental proof that it is a code. Again, the same story. You've got the experimental proof that there is a code, like in genetic code, and it's not. Okay? Remember this. No biochemistry books, the signal transduction is classified as a code. And then we have all the proof. We want to prove that there is a code. Okay, so this is the answer to the question which biology is. This is why the word code is foreign. It does not belong to biology. It's American. It's the way to think. Okay? One day, of course, people will realize that code is so false. But that will take, how many generations it will come. And eventually, what will exist, what does exist in nature, will exist in our books. Okay? And that will be the time in which codes will become Don't people be convinced that they do exist in nature. No. They want people to care about the situation, the theoretical situation on biology. And that's why code biology is a different kind of biology. Code biology is a biology with codes, which recognize the existence of codes. That's all. Okay? Right. Let's see a few other examples of codes just to give you. So, about splicing, yeah, I should have mentioned that. In fact, the rules of the splicing, we don't know. Not only we have the proofs that they exist, but we have the proofs that if they change, you got the names. There are lots of almost 20% and 30% of all these are changes in the rules of the splicing code. And those are the references. Okay? So we don't have the order book. We have to read all along, then you got the names. Okay. So now, this is another list. There is a list of codes. Genetic code, sickness code, signal transduction, balancing code, safety code, curing code, living code, random code. Okay. And for all of them, we can go through with the experimental proof that I showed you before. The story is simply the same. Okay? Two independent words, adapters, and unlimited number of possibilities. Right? We have this proof for all of them. Right? And then, for the first 3 billion years, the Earth has been inhabited only by microorganisms, and then visible form of life in plants and animals appeared only in the last 6 million years. And here too we have other codes. Several we have house code, region code, histone code, resolution code, apoptosis code, biological codes. And I repeat, we have the experimental proofs. This is not only just a list of names. Okay, for all of them. Okay. Neural codes. With the origin of animals, a completely new type of entities came into being. Entities such as action, potential, and neuron firings at the cellular level, and entities such as instincts and feelings at the organism level. Now, so, in addition to the organic worlds made of organic molecules, what came into being was a neural world made of neural states. Okay? We have codes between different entities, no longer between organic molecules, but between neural state, neural firings, or lower state neural levels and higher state neural. And here again we have codes. So the space code in the hippocampus. In the hippocampus make a map of the astronomy according to the rules of the code. And that, by the way, that was the 2014 Nobel Prize in medicine. Okay? So that code not only exists, not only have experimental proof, we have the Nobel Prize there to check, to prove that it is a real code. And yet people still do not believe that there are artificial rules in the biological system. Okay? So again, yes, it's a code, nice, but it's a word. It's only a word, it's only a metaphor. And this unfortunately, okay, then we have the neurocortical mechanism, all the acoustic, synaptic, olfactory, and so forth. Right? So this is the first one. When I said good biology is two things. It's first an experimental field of research. And the experimental field is this one. We expect a proof that there are many biological codes in nature. Not just one or two, 20. Okay? Throughout the whole history of life. And anytime new codes appear, pop, there's something new. Alright? And this takes us to the other world. If codes appear, if code exists, they appear in the history of life. And when they appear in the history of life, something new happens. So, isn't coding a new mechanism of evolution? Actually, the one mechanism brings novelties into existence. So here we go into theory again. Existence of many biological codes raises the right of theoretical problems, and in particular, the problem of the mechanism of evolution. This has been a highly controversial issue, as we know, and the great debate which accumulated in the 1930s and 40s, accumulated in the modern synthesis. The idea that natural selection is the sole mechanism of evolution. Now, okay, again, this is biology today. The sole mechanism of evolution is natural selection. Now, where does natural selection come from? Now we know that the copying of the genes is the elementary act of heredity. But when the process of copying is repeated indefinitely, another phenomenon comes into being. Because copying mistake becomes inevitable, and in the world of limited resources, not all changes can survive, so the process of selection is bound to take place. So, molecular copying in the world leads to heredity, and the indefinite repetition of molecular copying in a world of limited resources leads to natural selection. That's where natural selection comes from. The copying mechanism, mistakes occur, and then there must be selection of that because no changes can survive. So natural selection comes from molecular copying, and this means that natural selection would be the sole mechanism of evolution if molecular copying were the sole mechanism of life. If there was copying and nothing else, then there would be natural selection and nothing else. But the discovery of the genetic code has proved that there are two distinct molecular mechanisms of the basis of life. The copying of the genes and the coding of proteins. And the discovery of many other codes shows that coding is not limited to protein synthesis, but this is the word in many other cases. So if there was only copying, then we have only natural selection. But if there is copying and coding, which are two different mechanisms, they cannot be reduced to each other, then we must have two different mechanisms for evolution. Evolution by natural selection and by natural conventions. Okay? That is what biology is being up to. The other side of evolution, the other mechanism for evolution. Life, in other words, is not based on copying alone, it is based on copying and coding. And the existence of two different molecular mechanisms means that there are two distinct kinds of evolutionary change. Evolution by natural selection and evolution by natural convention. This is the main theoretical concept of biology. Okay? It's the other side of life. It's not only natural selection. There is the coding mechanism. There are two different molecular mechanisms at the basis of life, and that are big. There are different molecular mechanisms of which we have the experimental proof. evolution. So let's see if we go. So I don't have black things. Okay. So again, we say this natural selection is due to the copying of the genes, and it's copying of the uncoded so that the two different molecular mechanisms, right? The copying of the genes and the coding of proteins that implies that there are two different mechanisms of evolution by natural selection and evolution by natural invention. This is the major concept of evolution. Copying and coding are two distinct molecular mechanisms and two distinct mechanisms of evolution. Evolution is based on a mechanism and is based on a molecular mechanism, which is a evolution mechanism. Well, that's probably the greatest contribution of biology to biology. And the second phase of the second mechanism of evolution, which is actually the first one, because in terms of importance, is the coding mechanism that brings absolute novelties into existence. Natural selection changes, makes people change about pre-existing things. Codes become new absolute novelties, things that didn't exist before. Okay. And remember, once you have a code, then you must conserve it. You can get rid of anything else, but not the codes, because they already, it's like this. So, another theoretical consequence of code biology. Now, modern biology describes the cell as the system of genotype and phenotype, software and hardware. So a biological computer. The problem is that a computer receives its instructions codes from an operator which is outside the computer, an external codenator. Where this cell produces its own code, so it must have an internal codenator. So it cannot be a duality of genotype and phenotype, that's software and hardware. This is why good biology describes the cells not as a duality of genotype and phenotype, as a trinity made of genotype, phenotype, and ribotype. The ribotype is the ribonuclear protein system that is the function of the codenator of the genetic code. Alright? You know that the genetic code exists, and you know that there is an apparatus that implements the code. And that apparatus is the ribonuclear protein system of the cell. And that has nothing to do with genotype and phenotype. It's just the production of the code which is given to generate in order to work. Okay? The cell doesn't receive codes from the operator, it builds them from inside. So the cell is it must be free state entities to make a cell. Another concept, third theoretical concept. Okay? Another major biological concept is the difference between prokaryotes and eukaryotes, between cells that do not have a nucleus and cells that have one. Now, the prokaryotes or bacteria are continuously evolving and adapting to the environment, and yet they have not increased their complexity in the whole history of life. That's the point. Let's take the bacteria as a typical example. It's organisms that exist continuously, they change continuously, they adapt to anything, and yet their complexity does not change. And I wanted to, I have two quotations in order to give you some substance of this concept. So let's look at this one. The fossil record has revealed the presence of fossilized bacteria in most Precambrian rocks. The song that is stromatolized built by cyanobacteria 2 and 3 million years ago.
other words, appeared very early in the history of life, and that conserved their complexity in terms of size, shapes, and number of components ever since. So we have the problem with this complexity in terms of biology. Another quotation is one point. The bacteria have barely changed in 4 billion years of evolution. They are being matched in by natural fuels at that time. The rise of the oxygen of the air, the oxygen transformed in elemental opportunities, but the bacteria remains unchanged. The occasional global scale snowballs must have pushed the ecosystem to the brink of the blood, yet bacteria remain unchanged. There is nothing more conservative than bacteria. That's the biological reality. But the eukaryotes, on the other hand, have increased their complexity throughout the history of life. And this gives us a real problem. How is this possible that some cells could not increase their complexity and other cells became increasingly complex? And the solution to this problem is suggested by the codes in those cells. The bacteria never evolve new codes in the remaining bacteria. They remain at that low complexity. The eukaryotes evolve new codes, they become increasingly complex. Okay? So, the complexity of the system is determined by the number of codes. Right? That is another conclusion of good biology. It tells you what complexity is. If you want to increase the complexity of a system, bring on a new code. If you don't, you don't. You don't become more complex. So, let's go down to the nitty-gritty and say what biology is. And there it is. Of course, the complexity, well, let's just summarize what I just said. After the genetic code and the signal transduction codes, the prokaryotes have not evolved any other code, whereas the ancestors of the eukaryotes gave origin to many other codes, like structure codes, and codes, and the certain codes. These experimental facts suggest that prokaryotes did not increase their complexity because they did not evolve new codes, whereas the eukaryotes became increasingly complex because they continued to evolve new codes throughout the whole cellular life. And that's another fact. You don't have other new codes in bacteria, and they remain with the same complexity, 3 billion years, not just 2 days. Okay? If you want more complex reason, go and look what they have, and they have got new codes. These are experimental things, I repeat. It's not just theoretical deductions. Okay. So, I wish there was a way of switching from one. So, here we have 4 major ideas of co-biology. Oh, before summarizing that, I mentioned 3 ideas now. The fourth is the fact that another ideas of modern biology is that autopoiesis is the basis of life, which according to co-biology is not true. Because autopoiesis requires biological specificity. Autopoiesis means that the fact that the system has the ability to build its own components in all the sentiments. Okay? In order to build the same component all the same, you have to give them virality, and then they have to use the same rules in order to build the same proteins. So they must have the same code, the same genetic code. Without the genetic code, there is no biological specificity. So, autopoiesis requires biological specificity requires the genetic code. So the ancestral system that came before the genetic code could not have been autopoietic system. Okay? There is no way around it. The living system, the ancestral system, gave origin to modern life, could not have been autopoietic system. It
but they were co-creating systems because they evolved in the genetic code. So that's what's really fundamental in life. It's co-creasis, not post-creasis. Right? So that is the third concept. And so we got the four major ideas of code biology is evolution took place by natural selection and by natural conventions. The cell is a brain, genotype, phenotype, ribosome. The complexity of the living system is determined by the number of its cells. The fundamental process of life is co-creasis, not post-creasis. Okay? And I repeat again and again, remember, all of these statements we have experimental proof. Now, let me talk about a bit of the history now. The most important concept of code biology, the idea of evolution, created by natural selection and by natural conventions, appeared for the first time in 1985 in a book entitled The Semantic Theory of Evolution. The book underlined meaning is a fundamental component of life and propose that biology should become a semantic biology. That was the first name called biology, semantic biology, because in 1985 first of all many codes were not yet discovered. And the main concept was to introduce meaning in biology. So semantic biology is that, the biology with meaning, recognizing meaning is a fundamental component of life and biology. The term semantic biology is actually a theoretical position that I had from 1985 to 2003, and it also appeared in a book in the Organic, in a book called published by Cambridge University Press. The title was The Organic Code, an Introduction to Semantic Biology. So that was my position until then. But in 2001, Thomas Sebeer invited me to join the new field of Biosemiotics. And I totally accepted it. But in 2004, I gave up with semantic biology. And I got with Biosemiotics. Okay? Because at that time, I did believe the reason that semantic biology was a biology, whereas Biosemiotics was supposed to be more than that. It was supposed to be a union of biology and semiotics, a synthesis between the science and the humanities. And that was a good idea, that was a good concept. So I said, okay, so let's make biosemiotics as a new field of biology. Actually, I founded the German Biosemiotics. And I invited all people who are semiotic to join under the Biosemiotics. But there was a problem. Biosemiotics maintains that any semiosis is a process of interpretation. And it is an experimental fact that interpretation takes place in the brain. The brain doesn't interpret what happened in the book. But can you extend it from the brain to the cell? Now, protein synthesis is based on the genetic code, and any code is a semiotic process, which means that according to biosemiotics, there must be interpretation in code in protein synthesis. This amounts to say that a ribosome scans and messenger RNA and interprets this information. But this is precisely what does not happen. The codons of the messenger RNA are recognized by complementary codons of the transfer RNA. And saying that the ribosome is interpreting the messenger RNA is like saying that the key is interpreting a lock in order to open the door. And that's absolute nonsense. And that was my class with the biosemiotic chiefs. They insisted there is interpretation in protein synthesis, and I said, as long as you go, if you believe that, then there is going to be no biologist who will accept that. Because we have no evidence in the
you will remain an exoteric method of life. So, and that will be in time, and then we can crash. And actually, the governing mode of biosemiotics insists that the genetic code must be approximately interpretation. And it's something that we not accept. Now, many people say, I broke away from biosemiotics in order to find core biology. The truth is different. They threw me away from biosemiotics. There was an official, an assembly vote in 2012. They were asking for the vote for a directory of the biosemiotic system, one without and one with my name. And people voted for and they voted without my name. So it was thrown out of biosemiotics. So I had to find something else, and they found it called biology. Okay, that's the history. That's how the history went. There is no way that I wanted to run away and broke away. I could accept disagreement and so forth. But in the end, when you're thrown away from something, then you have to do something else. That's the evidence. Okay, so that's how core biology was founded. It was founded 6 months later, in the late month, November 2012, in Stellenbosch, where I was with Jan, becoming there. And then I went back to Italy and they put the constitution before a lawyer, before a notary. So it was officially there. So, and the term semantic biology was replaced by code biology because it is codes that creates meaning. It is a code that creates a role between the sign and the meaning. The object of two different words. So codes actually, the concept of word is rosary priority of the concept of meaning. In the beginning, this poem is semantic biology means that we have to introduce, as we did introduce information in biology, we have introduced meaning. But then on second thought, yes, in order to introduce meaning, you need a code. So the basic point is the code things. That's why the name became code biology. But remember, at the beginning, the first was in fact in the second one of therapy in 1985, before biosemiotics was ever born. Well, the biosemiotic as we intend today, the biosemiotic from Sibyl, implemented by Hoffmeyer, Chomsky, and Marcus, that guy there. Okay? Remember that in 2004 we went to Florence and so forth. Drank a lot of beer when we were around, we decided to find it in a way of the that's how it happened. And then that's it, that's happened. So it also Marcus was throwing away from this. I mustn't pronounce it. Anton is okay. And the actual terms biosemiotic appeared in print only 1991, 6 years after the publication of semantic biology. So it's not true. Semantic biology, code biology came from biosemiotics the other way around. Okay? Now, that was my experience with the biosemiotics. Now there is another experience. There is new movement. People say, let's go back to biosemiotics, or let's go back to biosemiotics. And that's another thing. That's a problem for a new generation to decide if the two can go together or not. Okay? The experience of the past has taught me that there is no possibility of having a dream, but who knows? Now the new generation of people here, you start, you want to connect semiotics with biology. You want to find the bridge. You want to find the biology. And how do you find it? Well, there are two different ways. And And this discussion, that's can all the way into the new world, and that's good, that's beautiful. Okay? And whatever happened, God knows what happened. But we will see. Okay? I told you what happens in the past. Nobody knows what will happen in the future. Maybe they do, but we find it. Okay? Now let's go to the concept of interpretation, which is the hard point of the dispute. What is interpretation? One might have the impression that according to core biology, meaning is always produced by colours. But that is not true. The best example of a biological process where meaning is not created by colours is a mechanism of interpretation. Let me give you examples. Animals can give meanings to the signal from the environment with neural codes, but some of them have also evolved means of interpreting what goes on in the world around them. And the difference between codes and interpretation can be illustrated with a classic example, which is sort of every time I said because it's beautiful. When a snake chases a prey, and the prey disappears from sight, the snake stops chasing. No prey in sight, no chase. When a wolf chases the prey, and the prey disappears from sight from behind the tree, the wolf goes on chasing. The snake is using the fixed rules of the code, whereas the wolf decides the prey still exists even if it is no longer in sight. The wolf makes a mental jump beyond the appearances, and this is what interpretation is. Okay? This extrapolation from limited data, this jumping to conclusion, is an operation Chomsky called abduction. And that is a fantastic example, is a marvelous concept. It's the third logical concept after the two introduced by Aristotle. After induction and deduction, first introduced abduction. So it is an important concept. It is a biological, and also we have also experimented with artificial neural networks that can jump to conclusions without enough data, and that abduction. So, Charles Peirce was a giant. The concept of action is an extremely important concept. It's compared to Aristotelian concept of induction and abduction. The point of the Biosemiotics is to make the measurement. That's the record. Okay. They want to purse even in the cell, the interpretation in the cell, and that's going beyond all logical things. That's it. So, abduction is a new logical category. The ability to interpret the world is based on that category. Interpretation is a form of semiosis because it is process that gives meaning to something. But it gives meaning to something because it is based on abduction, not on coding rules. So it's a different form of semiosis. Okay? So if semiosis giving meaning to something by codes, or by interpretation, which is a different process. So you have these two different. And code biology is actually about all processes that produce meaning, not just about codes. So it is about codes, but it's also about interpretation, and it's also about language. Language is another form of creating meaning. All right? So there are three types of semiosis here: codes, interpretation, production, and language, or cultural. And the origin of the world. It's about the origin of life, it's about the origin of mind, it's about the origin of language. Anything that produces meaning is about biology, and biology is interested in that. Right? And then I think we have, again, a summary of what interpretation is. Interpretation. So, many animals are capable of interpreting the hierarchies in the worlds around them, and the functional interpretation is not based on codes. It is based on a process that Chomsky calls abduction, a process that is different from the classic categorical induction and induction used by Aristotle. Abduction is a mental job, an intuitive conclusion, a sudden decision that we want to choose. Okay? Reaching a conclusion without enough data. And that has been done in the laboratory. They build artificial neural networks. And when the artificial neural networks are complex enough, they can give you an answer even without enough data. Solve this problem. These are the data for the problem. Reach a conclusion. A machine, if you don't give enough data, machine doesn't do anything. Those methods, some neural processes, they do give you a conclusion. Exactly. That's another example of abduction in the laboratory. Okay? Again, experimental reality. We have a correspondence in these diagrams. So, interpretation is evolved semiosis because it gives meaning to something, but it is different from coding because it belongs based on rules of the code on processes of abduction. That's it. That's what interpretation is about. Second form of semiosis. Now, then, in addition to the world of life and the world of the mind, then evolution gave origin to the third world of objects, a world made of tables and chairs, books and computers, houses and cathedrals. It is our world of framework of culture. And even in this case, we know that it is based on culture because we made cultural culture. There are conclusions of three kinds of objects in life: organic, neural, and cultural objects. And they were the results of three great innovations: the origin of organic semiosis, the origin of animal semiosis, and the origin of human semiosis. And this fit very nicely with what Open We're one, we're two, and we're three. Actually, for me, it's not one. And that's it. And to the three words, they correspond three kinds of semiosis. Okay? So that's called biology. It's larger than just codes. It's everything that produces meaning. Okay? So you see that word for the human semiosis, coding, interpretation, and language. So language is a new form of semiosis, different from all right. Now, it has been suggested that the reconciliation between biosemiotics and code biology is possible, because code biology originating from biosemiotics could easily go back, and this is a completely wrong conclusion. I think it didn't go at all that way. The truth is that biosemiotics and code biology has always been, since the beginning, two different fields of research, and this is demonstrated by the fact that the four major ideas of code biology, none of them exist in biosemiotics. None of them. So the difference is clear. Is it possible to have an agreement at the past? I don't know. That will be, it's up to the future. But the difference is that there's some complete difference, which is a real one. The split, anyway, it is because code biology is committed to the scientific study of life and being. And this goal has been explicitly written in the Constitution. It says that code biology study all codes of life from the genetic code to the post-translation with standard method of science. Code biology is a science and must use a standard method of science. There is no way in which you can accept any imposition of a concept from philosophical or whatever, metaphysical reason, etc., etc. So, yeah. Code biology and biosemiotics have in common the idea that semiosis is an integral part of life, that the semiotic process that we observe and exist should become an integral part of biology. And that's the big thing, to introduce a link between semiotics and biology, to show that semiotics is the biological reality before being the cultural reality. And this common goal is clear. That's the unification of science and humanity. It is that's common because semiotics and biology, but it is implemented in two different ways. At the moment. Right? Code biology analyzes the cause of the most elementary form of semiosis, and though we need to introduce semiosis in biology to show the experimental evidence to prove the existence of the cause in the living system. According to code biology, semiosis exists in the living system because cause exists in the system and cause are semiosis. But semiosis said, no, cause are not semiosis. And that's true. Let's call it irrational logic. Our system is given a completely different, supported by semiotics. I repeat, claim causes are necessary but not sufficient for semiosis. Semiosis exists only when interpretation exists. In protein synthesis, there is semiosis not because there is a code, but because the ribosome are interpreted. This is the end of semiosis. Okay? Let's be clear. If you want to bring biosemiotics back, you have to bring it up again, as it was in the original form of theory. It didn't use any of this. And I'm always trying to use those symbols that I was all right. You see, the biologist can accept that there is a code, but we never accept that there is interpretation. There is no mistake. And this is aggravated by the fact that different schools of biosemiotics have proposed different definitions of interpretation, and you work at the same time. Oh, but if you define interpretation in if you have a third, the fourth, and fifth definition of interpretation, you've got just a fifth. There's more confusion and nothing else. Okay? You add confusion to the confusion to other confusion. You don't solve anything. But if you define semiotic interpretation this way, then you can find no way. There is no way you can solve a problem by defining things in a different way. Okay? You can solve problems with links to reality, not by tricks of words and so forth. So, yes, again, it must be underlined the codes are not the sole form of semiosis. Wait a minute. They have been the sole form of semiosis for 3 billion years. For the whole of the history of life, of the great majority, codes have been the only form of semiosis. Only when animal came in, and we get this sort of interpretation-based mental semiosis, human semiosis. And non-English language, we got the culture, the third form of things. So, for the most part, semiosis is codes, for the most part of the history of life. At the end of the evolution, the time of the evolution, there are 2 different forms of semiosis, but they are simply mechanisms to give meanings to something. Okay, different mechanisms for doing the same thing. So, code biology concluded not only study code, it study all processes, the origin of the novelties of macroevolution, in particular, the study of the origin of life, the origin of mind, and of the origin of language. And I think that there, and that is it. That's what we have. Okay.
I wanted to ask about these 4 years and how they are compared to the Biosemiotics. So, you said both 4 years are excellent in Biosemiotics. As far as I understand, the main point is this interpretation. That's the problem. But not about these 4 years, how they are unfit for Biosemiotics.
Well, Biosemiotics has never suggested a mechanism for evolution compared with code. They just said semiotic things just happen. But in order to be a biologist, you have to make a precise mechanism. You know, the mechanism of coding is a specific mechanism you can find. And that semiotics never said that. There is actually a paper in which the biosemiotics list their basic ideas. You know, where was it? The biologist, something like that. You were not there. It was the other guys. They did list the basic ideas of biosemiotics and did nothing like that. So the fact that they are different is absolutely. See, the problem at the beginning when they started, let's try to put different schools together and try to find something, you know, that is linked to substance. And they did not succeed. Well, that was my favorite. But in my opinion, I did not succeed because they didn't want it. But the reality is they didn't suggest any new mechanism of evolution. So how they explain evolution is not explained. But what they always do, coding is a mechanism, it's different from coding. You can test it. And not only you produce codes, but you produce code and you must conserve it. That's actually the hard goal. Produce code and conserve it. Anything else will change. So that tells you what codes are. Okay? Yeah.
Thanks a lot for your presentation. May I ask you 2 questions about, first, about preservation of codes? Because in Karl we have a lot of mechanisms of preservation codes, and they are some system of codes by themselves. So the system of preservation of codes are in Karl.
Yeah, we call it conservation codes.
Yes.
We don't know how, but we know it exists. The codes must be conserved. The only thing that exists is the code. What is the mechanism? There we are going down to the nitty-gritty. Basically, we know there are mechanisms. If you change that sort of improvising into a different protein, you get cancer and so forth. They're all sort of molecular mechanisms. As soon as you step out of that, all the cancer. So basically that, if a cell trying to change the code, it gets killed. So far as we know is that. All experiments trying to change codes are at the end. That's the cell start. All the others. That's basically the mechanism of conservation. If you don't obey these rules, you get killed.
Okay. From this point of view, of course, in culture, we have culture memory, we have human memory, etc. So there's some mechanism which can be compatible with memory because if it's projectile or the wrong. So it's something like interpretation because you're right or wrong. So something in this case it's actual. It's actual in this case. It works. It has only two types of interpretation: wrong and right. If the sign is
No, wait a minute. About language or cultural codes, there's different forms of semiosis. It's not just interpretation, it's something different. And that's another thing. That's involves also in language. So let's begin to discuss in which ways language is different from interpretation and codes. Okay, because the human brain can interpret the word and can use codes and so forth. But language requires something else. And then we discuss what is it? Is it something else? And why is that language appears only in our species and not in other animals? All animals have ways to interpret the world, all animals have codes to interpret the world, and that's why we read the whole language. So that's why it's not enough to have interpretation. All animals interpret the world. Okay? So there's another form of semiosis. What is it? Where does language come from? Huh? No, not at all. Language, not at all. All you have now, in the cell, right? You have the adapters, which are the transfer RNAs. In language, you have that, the code, you have adapters, and the adapters are human beings. That's the difference. To have molecular adapters and to have human beings as adapters in the code. But that's the big difference between the two language semiosis. Is in the adapters. Okay?
we have pragmatics. That is a system of
Could you
analysis.
speak up? We can't hear.
Okay.
We can't hear the question.
Sorry.
Will you to stand up, look up, take your best? Yeah, you're right.
Yes.
He's always stand up.
Okay. As Professor, some questions about preservation, of course, preservation mechanism. How it is possible to interpret this mechanism as an interpretation, maybe in the case of some censorship, when we distinguish wrong and right sequences. And finally, is it possible to think about a code maker as an interpreter who produce the codes and who decide what code is wrong and when it should be deleted? Okay. And thank you for the question, because it's a lot of food for thought.
The difference is in the adapters. Think about it.
Yeah, but in language we have some system of adapters, we call it pragmatics, which provide the interface between the language as a system and our communication. So we just find something similar.
Oh.
Maybe.
Marcella, after.
Yeah, you choose that. You want the microphone? Otherwise, Armin is the right people for playing.
Should be okay. Can you hear me?
Yes.
Okay.
They can take that, otherwise.
Thank you, Marcello, for your talk. Always a pleasure to hear from the source. I'm just wondering to what extent you admit that from the basic organic codes, like from genetic code on, codes must be degenerated, signs must be polysemic, and meanings must be selective in order to keep the ability to take into account contextual keys and an open-ended possibility for evolution. And this is not interpretation.
Yeah, I think, it's okay. Yeah.
Do you admit that a code, even though it evolves by reducing ambiguity to some extent, it does have to keep ambiguity?
Yeah, and so far, as we know, is that the only mechanism is that any cell could
the gates of the code get scaled. That's the mechanism we know.
Yeah, but the code is not a one-to-one rule. Otherwise, it's not a code.
Oh, yeah.
Okay?
Yeah.
Okay.
If you, you can find the code only if you find a potentially unlimited number of rules. Then you have to do this code.
But not, what I'm saying is that not, it could have been otherwise. But being the way it is, it's not a one-to-one rule. It's a degenerated way of standing for things.
Yeah. You know, I used to
It's
say.
very happy. Did you know that?
You see, life begins. You have all sorts of molecules in the world, and some molecules are formed spontaneously. Then you got a few molecules that started producing bonds between other molecules. So they started producing artificial molecules. Okay? And that's where life started. When you have molecules produced not by natural processes, but by machines. So artificial molecules. We used to say, life therefore is a making of artifacts. Proteins are artifacts. And if you shorten that, you end up with the word life is art. Okay, that is short for artifact making. So, do you have rules in art? Certainly in art you have the rules that not everybody gets killed. If he doesn't break the rule, I think that's another question. But that's it. The invention, there is no need to invent new things. It just happens. And then you have to survive. There is no necessity of bringing a code instead of the other. The bacterial lived for 3 billion years without inventing anything. Okay?
Sure, just wanted to clarify your answer. So you allow for ambiguity in codes, but do not allow.
Not ambiguity, I'm talking arbitrariness.
But you are looking about ambiguity.
Yeah.
You agree?
Well, what says you?
Marcello
Marcello.
There is ambiguity that has to be an abduction.
No.
It's more in the sense that there are multiple.
Ambiguous, a code is ambiguous. Before you have a genetic code, you necessarily have an ambiguous code, which means the code you can associate different kinds of organisms. That's ambiguity. But that's also arbitrary. Well, no, forgive me.
Don't do that.
No.
That was very.
I understand exactly what she said, even if she used the opposites.
Let's replace it for polysemic science. I'm okay with that.
Okay, yeah.
Science are still polysemic for the organic.
Yes, we have some context dependent.
Okay. Okay, let's keep ambiguity different from arbitrariness.
Yeah, definitely.
They are different, polysemic and arbitrary.
Even for example, in the Morse code, you can have some kind of regularities. For example, the words, the letters they use more often, they associate with the smaller number of dots and dashes. That's the rule that we have there, but it's still, it's semantic very close.
I don't want to pose another question when you give someone wants to chat.
There's a question from Anton Marcus with Josh.
Anton, okay.
Two comments to the beautiful dialectics of your theory, natural selection versus natural conventions. Of course I do.
Okay, fine.
First is a comment to your question. Why writer codes are so conservative? Well, why the internet is so conservative? Because the protocol over the whole globe must be maintained. We all are members of a genetic networks by horizontal gene transfer, and nobody is going to change the protocol, otherwise he or she dies.
That's the point nature uses. If you break the rule, you're dead.
The only exception is mitochondria because they are
But remember, to kill a cell means to have a mechanism for killing it. the cell gets lost. No, you get killed by, there are specific genes it cause for cell series. So you activate them and the cell becomes.
Where this protocol is written? Where is the book where this protocol you can find it?
Well, it's then the first part, the natural selection comes. It's a physical force, let's say. Yeah, but no, the second comment, because I think the devil dwells in the details, natural conventions. So somebody must come to a convention. Something must come to a convention, must negotiate how the codes will look like. So who came to conventions? If I would be not your enemy, but no, if I would like to be to back to you, I say okay, but isn't it a seven of my please?
Yeah. Can I answer him?
Sure.
What was the mechanism that brought the genetic code into existence? That's what you want to know. Why do you
call it convention?
I call it, well, a convention is a code. It's another name for a code.
But I agree there.
Huh?
Convention
in my opinion, natural conventions is equivalent to natural codes.
I see.
Okay? A convention is something that you agree with somebody. The rules is this one.
But who made the agreement?
Yes. How did we code it?
I wanted just to say one of the mechanisms would be the minimization of the errors, for example, and we will speak tomorrow about this, for example, but we don't know for sure, of course.
Wait a minute.
But it's just a play of words, right? To call it convention, that's it.
Wait a minute. Don't. The term convention, in my opinion, as I said, is equivalent to code. It's something, it's a rule which is agreed with. Now the point is, how did nature invent the code? Why did they evolve the convention? The convention by the genetic code. That's the other problem. Okay? So how did the genetic code evolve? So, there are some RNAs that can stick, can build the peptide bonds between amino acid RNAs. So they can build proteins, statistical proteins. Okay? If you have an RNA that makes peptide bonds, make peptide bonds between different amino acids, you have a statistical. It's an amino acid that has any kind of sequence you want. Okay? So you can imagine a world of RNAs that made statistical proteins in order to stabilize themselves, in order to so on. So the ability of RNAs to produce statistical proteins is there, and they produce statistical proteins in order to have be more stable and so forth. So you start from that ability, which is again, it is true. I mean, there are RNAs that make peptide bonds. That's it. So what's the evolution of the system? Of course, they couldn't evolve the code because of the advantages of biological specificity that came in the future. There must be some practical advantage. And what is a practical advantage? But if you have roughly ribonuclear protein machines that build statistical proteins, what is an immediate advantage for those systems? Is to improve the machines to make statistical proteins. So in terms of number of components, if you increase the number of components, number of ribos, for example, the ribos become more stable to thermal loads. So if you want to avoid the errors caused by the thermal fluctuation, just increase the size. Wait a minute. Good. And then other things, if you want to transmit
different families of statistical proteins to the progenitors and so forth, you have to reduce the ambiguity of the original code. Okay? So that is an immediate advantage. If you want to transmit the ability to do the same statistical work from one generation to the next, you will increase the mass and you reduce ambiguity. Okay, reduce ambiguity, and you have biological specificity. That's how we make good things.
But
Without, there's no immediate advantage, but that's it. That's a logical example of a system which is based on this level of reporting machines, and the only problem is how to improve these machines to make them specific. They didn't think about specific. So the mechanism is there, it has been published. And the fact that you haven't read it doesn't matter.
Professor
yeah.
Barbieri, when you said that there is to reduce ambiguity, you are saying that it's like a system that are trying to reduce the chaos, the errors and the chaos.
One column can be associated to many nuances.
Okay.
Not just one. That's ambiguity. And at the beginning, it was like that. It must have been like that because there was no way. And then you have this evolutionary trend towards a system where you reduce the number of amino acids that are associated with coding. And in the end, you have only one amino acid. But you must have a mechanism that explains it. Why? Because it was in the interest of this system to give to the progenitors, to the successors, the same ability to produce the same families of statistical properties. They didn't think about specific properties. But the only way to enable the descendants to have the same kind of statistical proteins, you have to the trick is to reduce ambiguity. And in the end, you end up with this method. Okay? It wasn't written anywhere, it was a necessity. It just happened. Because the only way in which this system could survive. And are we going out of time anyway?
Yeah, so I propose we continue the discussion outside the coffee break. There was one last question from judges. She wanted to
Actually, he already answered it.
All right.
Yeah.
Okay. So we continue in 20 minutes.
Can you zoom out, please?